Greetings everyone. My name is Devlina Ghosh. I am a PhD student from Amity University, Lucknow, India. Today I am going to discuss about my research work, the topic of which is HMGB1 inhibition attenuates LPS induced neuroinflammation, cognitive dysfunction and sickness behavior. As we know that microglia cells are the primary innate immune cells of CNS, and they scan the microenvironment continuously during their resting state. Whenever encountered with any kind of trauma, they get activated and they facilitate the secretion of various pro-inflammatory cytokines, persistent secretion of which may lead to various neurodegenerative disorders. Now, high mobility group box 1 is a highly conserved nuclear protein and is released from glial cells in case of trauma. It activates the NF-kappa beta pathway and promotes the secretion of pro-inflammatory cytokines. Thus, inhibiting or blocking the secretion of HMGB1 could be a very lucrative target for therapeutic treatments in neuroinflammatory conditions. Glycyrrhizine, which is a natural extract from the roots of licorice plant, is an antagonist of HMGB1. Studies have shown that in case of focal cerebral ischemia, glycyrrhizine reduces the level of oxidative stress. However, the underlying mechanism behind this anti-inflammatory property of glycyrrhizine is still largely unknown. Based on that, in our study, we hypothesized that glycyrrhizine, which is an antagonist to HMGB1, may weaken the adverse effects of neuroinflammation, thus may improve the functional outcome. In order to do that, we have developed an LPS-induced neuroinflammatory model in vitro as well as in vivo, and studied the pro-inflammatory response in microglia as well as neurobehavioral function at different time points after the LPS stimulation. For the inhibition of HMGB1, we have used glycyrrhizine and analyzed key outcome measures of neuroinflammatory markers and neurobehavior after LPS treatment. So for my in vitro study, I have taken BV2 cell line, which is the murine microglia cell line, and have divided it into three groups, one with LPS stimulation only, another with LPS plus glycyrrhizine treatment, and the one which is control. After the activation of microglia cells, I have collected the conditioned media as well as the microglia cells to determine the key changes in free radicals like nitrite and the changes in reactive oxygen species level. As you can see very clearly and is evident from this graph, that when we compare the LPS stimulated cells and the glycyrrhizine treated cells, there is significant difference between the nitrite release, and we can see there is significant reduction in nitrite as we have increased the glycyrrhizine treatment, the amount of glycyrrhizine used. And we have also studied the changes in reactive oxygen species level, and as we can see here very clearly that the level of reactive oxygen species is much less in case of glycyrrhizine treatment when compared to LPS only condition. Moving on to the in vivo model, we have used the bulb-C male mice, treated them with glycyrrhizine and followed by LPS stimulation. We did the pro-inflammatory marker analysis and behavioral analysis. As we can see, the glycyrrhizine treated group after LPS stimulation shows significant reduction in the level of pro-inflammatory markers when we compare them to the LPS stimulated group without any treatment. Now moving on to the neurobehavioral aspect, we did the tail suspension test as well as the Y-maze test to analyze the changes associated with stress condition of the animal as well as the cognitive function of the animal. So as we can see in this graph very evidently that when compared to LPS stimulated only condition compared to the glycyrrhizine treatment model, there is significant difference between the stress, difference in the stress condition. And in Y maze 2 we have found that the cognitive ability of the mice has significantly improved in the glycyrrhizine treated condition. So in our study we can conclude that the LPS mediated activated microglia is definitely associated with neuronal dysfunction and gives rise to anxiety and depression like behavior. However, when we treat the same condition with glycyrrhizine, it reduces the level of anxiety and depressive like symptoms in our mouse model. Targeting the HMGB1 using glycyrrhizine could prove to be a very promising therapeutic possibility, and it can regulate the neuroinflammatory condition and may further improve the functional outcome. What do we expect from our study? So, this detailed study will definitely provide a strong platform to understand the regulation of neuronal function and underlying the risk factor in development of various movement disorders which are associated with various neurodegenerative diseases like Parkinson's disease, Alzheimer's disease. So our detailed study will also help to understand the mechanism underlying the oxidative microenvironment formation and also different dynamic changes which are associated with it. These are some of the papers that I have referred to during my research work. Thank you so much.